In this video, we'll be breaking down the process of transcription. Before we get down to the nitty-gritty details of transcription, let's take a step back. Why do our cells want to go through this process of transcription? It's all about protein production. Your DNA contains genes, and these genes have in their sequence of nucleotides the instructions for building a protein. There are two major steps for being able to use this information in your DNA to build a protein. First, you must make a copy of the information stored in the DNA, and then you must read that copy and build the protein. The first step, the production of a copy of the gene, is called transcription. The second step, the reading of this copy to build a protein, is called translation. So we're going to focus on transcription. We have a gene that contains a nucleotide sequence that leads to the production of a protein your cell needs. Our end goal of transcription is to make a temporary copy of this nucleotide sequence that will then move into the process of translation. This temporary copy comes in the form of RNA, which, like DNA, is made up of nucleotides, but unlike DNA, is a single-stranded molecule and contains the nucleotide base uracil instead of thymine. There are many types of RNA found in cells. The specific type of RNA that is built in transcription is called messenger RNA, or mRNA, because it's carrying a message from the DNA to the protein-building machinery in the cell. So how do we build this mRNA transcript of a gene? We need to use a few types of proteins. RNA polymerase is the enzyme responsible for unwinding the DNA and building the mRNA. And proteins called transcription factors are responsible for promoting transcription of a gene by recruiting the RNA polymerase to that gene. So if we look at a gene, we see that we have a promoter region, which is where all of the proteins necessary for transcription bind to the DNA to prepare for the process. Then we have the transcription unit, which contains the actual sequence that codes for the protein. And then we have the termination sequence, which signals the end of transcription. The promoter region of the gene usually contains a sequence called the TATA box, which contains the sequence TATAA. This TATA box in the promoter is where some of the transcription factors will bind to the DNA. Once the transcription factors have bound to the TATA box, they can recruit the RNA polymerase to the promoter of the gene, helping it bind to that region upstream of the transcription unit. So if we look at a gene in DNA, we see that there are two strands of DNA running anti-parallel. One strand contains the nucleotide sequence that has the instructions to build the protein. This is called the coding strand. And the other strand is simply the complementary strand, which is called the template strand. To make a molecule of mRNA that is a copy of the coding strand, the mRNA strand will be built complementary to the template strand of the gene. This way, the sequence of the mRNA is simply the RNA version of the coding strand of the gene. Once the RNA polymerase has bound to the promoter region of the gene, transcription can start. The RNA polymerase will unwind the DNA, creating a transcription bubble. The RNA polymerase will then use RNA nucleotides to build a strand complementary to the template strand in the transcription bubble. As the RNA polymerase moves along the gene, the transcription bubble will remain the same size, meaning that the DNA that has already been built onto will wind back together. For this to happen, the newly built mRNA will begin to detach from the DNA toward the back of the bubble. As the RNA polymerase moves down the gene, an mRNA tail will start to trail the bubble, getting longer and longer as transcription continues. When the RNA polymerase reaches the end of the transcription unit, it will run into the terminator sequence of the gene. This sequence of DNA causes the RNA polymerase and the mRNA to detach from the DNA and the remaining transcription bubble winds back together. Notice that the original DNA strand is left completely unaltered from its original state. At this point, the RNA polymerase has created an mRNA version of the coding strand of this gene, and this mRNA can now go through the process of translation to build a protein. And that's the process of transcription. Our cells use transcription factors and RNA polymerase to make an mRNA transcript of a gene, which can then be translated into a protein needed to complete a certain task within the cell. Please see my video on translation to see how this process goes all the way through to the creation of a protein. If you're interested in the genetic code, you can see my video on how you can translate mRNA into a sequence of amino acids.